So I'll first start with thanking the organizers for inviting me. It's a lovely set of talks. I really learned a lot, and thank you and congratulations, Deepak. I've been sandwiched between two turbulent lectures, and so this will be sort of. But I think that maybe the common theme might be finite time singularities. So. So what I'll talk about today is some recent work that we have done. This is in collaboration with my postdoc Ayan Rai Chaudhary and student Saptarishi, and it's on the emergence of singular lines of force chains that that emerges as a result of the active hydrodynamics of the cytoskeleton, the active cytoskeleton within the cell. So it's going to be some amount of biology, but I'll just have 2 slides on biology, that's all. So let me just define, show you the context. Okay. So this is a image of a eukaryotic cell like ours, and the cell lacks a cell wall like our cells. And because of that, the membrane is very floppy. Okay. But yet the cell has this cell is adhered on the substrate, sitting on a table. Yeah, and it has a very definite shape. Yeah, and a definite size. And what gives it its shape and size is predominantly these green lines here, which is called the actin cytoskeleton. Okay? These are rigid rods, which are distributed within the cell. It's called the actomyosin cytoskeleton, and these are active filaments, okay, because they hold tension. The moment you break these lines, the whole cell will collapse. Yeah. Okay. Now these lines here, since it's sitting on the substrate and it's adhered to a substrate like the table, these lines are held taut by anchoring sites on the cell surface. These are called focal adhesions. Okay. So what I want to talk about is the emergence of these sort of singular bundles of lines. Yeah, that's what I want to talk about. It's interesting that the moment you detach the cell from the substrate, yeah, peel it off, then this whole set of bundles immediately depolymerizes and breaks up. Okay, so there's the existence of these stress fibers. These are called stress fibers. The existence of these stress fibers is due to stabilization by these surface anchors, which comes about because the cell is attached to a substrate. Yeah. Okay. So is that fine? Okay. So now the same picture. I'd like you to see, this is what you saw here before. And this one is a phenomenon that coexists with this kind of structures. It shows the excitability of exactly the same thing, the actomyosin network. And so you see there's a coexistence of excitable waves with these singular lines, these stress fibers. And this is a question that we've asked. How do these local active forces that are generated by these structures, by actomyosin, result in a long-range dynamic force patterning? How do they simultaneously exhibit such singular stress fibers and excitability? Okay, so this is the question we ask. Yeah. This is all over your. Is the tissue sitting? So all yeah. So all these cells are attached to something or the other. They're not floating in blood like blood cells or something. This one? Yeah. So this is the same things. What you're seeing is actin and myosin. An image of which one? These ones? These are. I mean, there'll be also monomers and all these things. Yeah. So you fluorescently tagged these molecules, and most of them exist in these fibers. Yeah. Yeah. They're the same kind of cell, but they're two different. Yeah. This was done by Thomas Anton in NCBS. Okay. But this phenomenon is very general. Okay. It's that, imagine that I start off with a sort of uniform distribution of actin and myosin distributed uniformly on the cell, in the cell. It's now adhered to the cell substrate. It suddenly, after some time, settles into this kind of configuration of singular lines. But simultaneously, you see this dynamic excitable waves.
So
this one? This one? Is this in real time? May not be. It may be sped up by something. I don't remember. Yeah. It's in, but it's a live cell imaging. Is that? But the movie may not be in. It's on the order of seconds. This excitable media is of order of seconds. Yeah.
But I'll get to the real. I mean, that's
Yeah.
That's the time scale
Yeah,
of the.
yeah.
It's not if speeded up or slowed
Yeah.
down. Thank you.
where are the rods in the right field?
Well, see, this is one small section here. You know, so this is a much higher resolution.
So,
sorry. It's speeded up, yeah.
if I were to make a movie of the first picture,
This one.
would that show something, a lot of fluctuation, or will it be?
No, you'll see a lot of fluctuations. Yeah. These are very dynamic structures. They're constantly undergoing turnover, so binding and unbinding, and even large scale motion. Right? Okay. So at least the system is clear. Okay. Now I have to. These are two slides in biology that will tell you about actomyosin. Okay, because I need to describe that. Now actin is a rigid rod shown there, but it has a certain polarity. So it has a plus end and minus end. The polarity arises from structural polarity of the constituents of this actin filaments. This is a motor protein. It's called myosin. It consumes ATP and therefore moves towards the plus end. Okay, and by moving towards the plus end of actin, you see that the filament is pushed backwards. Okay, so this ATP-driven current is a consequence of the interaction between myosin and actin. Here is an ATP-dependent stresses that can be generated in the medium. And you can see here that if this myosin filament binds to these actin filaments up there, then this head moves towards the plus end, this head moves towards the plus end too. Therefore, it pulls the filament in this direction here and that direction, giving rise to contractile stresses. The same kind of argument in this configuration gives rise to extensile stresses. Okay, so in a random collection of these filaments and motors, you'll find both contractile and extensile stresses, but there is an asymmetry between the contractile and extensile things coming from both kinetic considerations, namely the speed of the motor as it comes towards the tip here gets slower, and also because of mechanical asymmetries. This one shows a buckling like so, and this one doesn't have that. So there's a difference between the extension and contraction, giving rise to a net buckling of a random network of this. So if I have a random network of filaments like that with a lot of motors, typically it will show this contractile behavior. Okay. Okay. So this is to say that a system of actin and myosin shows gives rise predominantly to contractile stresses. Is that right? And these are actively driven. They are ATP. They consume ATP. Okay, so this is one aspect. The actomyosin system is a force generator. It's a contractile force generator. Okay. In addition, the actomyosin system is actually a force sensor. Okay, and this is because of the following: that if you have myosin attached to actin and you apply a force, then it holds on stronger. Okay, so this is called a catch bond. A lot of molecules in the cell do that. It's like a seat belt. The moment you push forward, it holds you back. Okay. Yeah. Does it unbind? Yeah. Oh, I see. No, that's so before it does that, it'll break. So what usually happens is the filaments break up. Yeah. Huh? The persistence length of this is very high. It's not like a rope. So the persistence length of these filaments is of the order of the cell size. So they're pretty rigid. Yeah, so for instance, thermal fluctuations won't easily bend this thing. But of course, here the stresses are coming from activity. And so they bend. But if you bend them too much, they just break. Okay. So experiments have shown that if you look at the bond lifetime, the bond between the myosin and the actin, then typically it shows this catch bond behavior in this certain myosin species called myosin 2A. Okay, that is the bond lifetime increases as you apply a force, so it holds stronger to the actin. Yeah, but in some cases you could also have a slip bond. In some species of myosin, you could have a slip bond. That is, it gives way. Yeah, it unbinds very quickly as soon as you apply a force. Yeah? Okay, so you could have these two characteristics. I just wanted to say that the binding of myosin onto actin depends on force. Okay, it's force dependent, strain dependent. Yeah, so these are the two features that we're going to put into the theory. Actomyosin is a force generator, a contractile force generator, as shown here, and actomyosin is a force sensor. Okay, and in fact, this is going to be the physics behind force generation and force sensing is very different, and so so this is the reason why a lot of this peculiar features having to do with broken time reversal symmetry occur. Okay, so it's because the force that it generates in the medium is not the same as doesn't come from the same physics as the response. Okay. Okay. So here's the question that we want. We start off with a cell where it's an isotropic actin mesh, myosin uniformly distributed in the cytoplasm. And then we want to ask whether we start time t equals 0, you'd put in ATP. Does it eventually form these kind of singular lines, these bundles of lines, okay, because of the interaction between actin and myosin. Okay, so we've worked on this in a variety of models where you have either one species of myosin, and this segregates into myosin rich and myosin poor regions, or you can have two species of myosin, which I'm calling myosin A and myosin B, where the segregation happens between myosin A and myosin. Okay, but I'll just talk about one theory because the results are the same yeah, all over the place. Yeah, so this is put in the bath of. That's an interesting question because you could, for instance, imagine that you're localizing this and ask how does the system respond? No one has studied this, sorry. But that's a more difficult experiment. Say that again? It's not an equilibrium process. Yeah.
Because of the extra force you are applying?
Right. So there is yes, exactly.
Even then, it's not just with the force. You cannot just think of it like a Kramer's.
Yes, absolutely.
Then it's equilibrium.
That's equilibrium, right? So in the absence of any force, then there is a binding and unbinding, which is very, very, very slow, okay. And that is thermally driven.
But even with the force, now I cannot just think of as a Kramer's.
No, because the force is also coming from the activity.
Force is also coming.
Yeah. But we'll model the unbinding and binding by a Hill form. Yeah, so it's like a Kramer's with a force. Yeah,
that's an equilibrium process, right?
Well,
in the sense that
that's an equilibrium form. Okay, let me go to that and then we can discuss this one. Then you can decide whether it's equilibrium. Okay, so I've said this. Yeah, so this is the hydrodynamic theory you'd like to construct. You have a mesh of initial mesh of actin. We'll assume that it's an isotropic mesh. And so locally, you can describe the mesh by a displacement field U, which is a function of space and time. And from here, and thereby define a strain, which is this, which is the symmetrized derivative. I mean, epsilon ij is just diuj plus djui. There's a strain. Epsilon here is the compression, is just the diagonal part of the strain tensor. And given a strain, of course, you have a stress. The stress has three parts. One is an elastic part, which will assume linear elasticity theory, okay, to start with. Then there is an active contribution, which is a function of the actin density and the myosin density. And then there's a dissipative part, which is just the viscosity into the strain rate. Okay. So on the right hand, on this equation is merely force balance. It says the local mesh velocity is equal to the net force. And the forces come from internal elastic forces, active forces, and the standard dissipation, viscous dissipation. Okay, so that's that equation. And that's the sigma active is where the active physics goes in. Okay, the rest of the things are purely thermal passive terms. Next, you have myosin, which binds and unbinds. So the binding and unbinding of myosin. So myosin is advected. Bound myosin is advected by the velocity of the mesh. There is potentially a diffusion term, which will take to be small. And then there's a turnover, namely the binding and unbinding, which is strain dependent. Okay, so these are the set of equations that will be. Okay. Now, as I said, the elastic stress is merely, we just start off with a linear elasticity theory. So the stress is proportional to the strain with an elastic modulus B and U. Okay, this is the Lamé coefficient and this is the shear modulus. Yeah. Okay, so this is just that. The actin mesh is, of course, not only defined by a local displacement field, but also a density field. But the density, we take the density field to be a fast variable. So the density is slave to the local compression. Okay. If I locally compress the meshwork, locally the density of the mesh will increase. Okay, so that's what this tells. Yeah, remember, for compression, epsilon is negative. Okay. Then, having described the way the stresses appear, I haven't discussed the active stresses, but just the passive parts. Let's look at the turnover. So the turnover is a combination of binding and unbinding, and we take the unbinding term to be strain dependent, and of this hill form. Okay, it has a bare part plus an e to the power of epsilon, epsilon being the strain. Yeah, and the sign of this alpha tells you whether it's a catch bond or a slip bond. Catch bond is where it holds onto it when the force is applied. A slip bond is where it releases it when the force is applied.
So rho A was the mesh density, and then rho B was the myosin,
myosin density. Yeah,
and
yeah.
what is rho I?
Rho I are the two myosins. Sorry.
Oh, true.
Okay. Yeah, I'm sorry.
These are two species of.
Two species of myosins. Yeah, yeah. So rho 1 and rho 2 are the two species of myosin.
In the previous equation.
Yeah, I wrote it as rho B. I'm sorry.
Rho B. Oh,
Yeah.
so you have two equations for the two.
Yeah. So remember, I said that. Yeah. There are two situations I could look at. One is I only have one myosin species, and the segregation will be between myosin rich and myosin poor, or I have two myosin species, and the segregation will be between myosin A and myosin B. Okay, so I'm just going to have, let me just write one theory here. Yeah.
So Madan,
Yeah.
Yeah, in the next slide.
Yeah.
Two confusions. One is, I thought the catch bonds were the ones where they held on stronger.
Yeah.
But that would mean that the unbinding rate should go down, right?
Remember, epsilon is negative.
Oh, epsilon is negative.
Yeah.
Oh, thank you.
Compression. Yeah. Yeah.
Okay.
Yeah. Sorry about that. Yeah.
That's all right. The second is, it is, I mean, for the catch bond, this was non-monotonic, which you're not dealing with.
So there could be non-monotonic behaviors in how the unbinding depends on the strain. We haven't included that yet. Yeah. Okay. Yeah, okay. Now, it's clear that if you write down equations for U, rho1 and rho2, which looks similar to that, I could also always go to, phi and rho, which are the sum and difference variables, the segregation and the net density. And these are the set of equations. So these are the set of equations that we're going to analyze later. Okay? And what we're going to look for are segregation instabilities and so on. Okay. Now, I haven't told you anything about the active stresses. The active stresses, as I said, was depended on the density of actin and density of myosin. And the way we do it is just to decompose it in this manner, okay, and expand the actin density about actin density is slave to the compression, right? So delta rho, the excess density, is proportional to the compression. Yeah, and so I'm just going to expand this in delta rhos. Yeah, and you get this whole bunch of terms here multiplied by the dependence on myosin density, which is there. Okay? So note that we're just going to take the isotropic part of the stresses in this case. This is scalar theory. Okay? Now, if you unpack this and separate terms, what you find is that this active part renormalizes the elastic stress. Okay? Initially, we just had an elastic stress where sigma was proportional to epsilon with a coefficient b. Yeah. Now, because of this nonlinear, unpacking, you find that there are terms that are proportional to epsilon square and epsilon cube. So you generate nonlinearities which come from this contribution. Okay, and the resulting stress actually has this nonlinear dependence on epsilon. So what started off as being a linear elasticity theory now becomes nonlinear because of activity. Okay, and what you find is that because of the sign of this B2, it could be negative, and so you get two minima here instead of the single minima linear elasticity theory that started off. Okay, so activity renormalizes the elastic free energy to generate two minima. Okay, and you'll see that one of the minima exists at negative epsilon, which means the compression region. Okay, so you'll find that as a result, myosin likes to move towards regions which are more and more compressed. Okay?
Madan, these equations are completely deterministic, right? You don't have any thermal noise.
No thermal noise. Yeah.
Do they play any role, thermal noise?
I think the active stresses are much larger than KT, so that shouldn't. Yeah. But you could ask what happens if you change temperature, and then that's a dangerous thing to do in the cell because hell breaks loose. Many things are temperature dependent. Unbinding, binding is temperature dependent. Chemical reaction rates are temperature dependent. So yeah. Okay. There is a spatial dependence. Yeah. I'll show you that. Yeah. I just before doing the calculation, I just wanted to show you what are the interesting features that. This is a mean field. Yeah. This is a mean field and it's a local.
How many parameters do you have dimensionless in this model now?
many parameters, but we'll see what we can get out of it. Yeah. I mean, the idea here will not be to make quantitative comparisons with experiments. Experiments don't exist so far, but just to get the qualitative behavior. Okay, so I just want to get back to this. Remember, we started off with a theory where sigma, the elastic stresses, was just proportional to epsilon. And now, as a result of that unpacking of the active stresses, you got this a huge slew of terms, which included nonlinear elasticity theory, okay, and a renormalized B, renormalized elastic modulus, okay, and the existence of a back pressure. Okay, so the back pressure sigma 0, yeah, is proportional to the so it's a local because rho and phi are local. Yeah, so the back pressure is also so this is the form of the local this thing. So you get an active contribution to the back pressure. You also get a renormalized elastic modulus, which is the coefficient of the linear term there. And it's given, this is how the renormalization happens. This is the bare elastic part, the linear elasticity theory. and that gets reduced by this extra term. Okay? So you can imagine that if now there's a concentration of myosin in one region, that will increase this quantity. And so this can be driven to 0 and negative values. Okay? So you get a potential instability coming in here. Yeah. In fact, this material is really interesting. Overall, the system is not unstable, mechanically unstable, because there is a nonlinear stabilization term. Okay? But this is just that this linear part appears to have an instability. Okay? This gives rise to a negative Poisson ratio. That is, if you pull it here, it can expand in the perpendicular direction. Because the Poisson ratio is just the ratio of this B minus mu divided by B plus mu, okay? Where B is the bulk modulus and mu is the shear modulus. Okay? So these have very strange elastic properties. This is what I. The other how much time? Oh, not much. Okay, so maybe I'll skip what I was going to say. But okay, I was going to say that this drives the elasticity to be hyperbolic rather than elliptic. Okay. Now, so here's the segue. So now one can do a linear stability analysis of those set of equations that I showed you. The equations in phi, segregation order parameter, rho, and displacement field. And that eventually gives rise to a phase diagram. Okay? And I'll just plot the phase diagram here in terms of the relative contractility, that is the difference in the contractility between species 1 and species 2, myosin species 1 and species 2, and the relative unbinding. Okay? Because these two myosin species could provide different contractile stresses and can unbind at different rates. Okay? So this is the relative. And you find that in a large region of parameter space, the dispersion curve, the growth of the instability as a function of wave vector q looks like so, okay, which indicates an instability. So this is the classic spinodal decomposition that you get in. Yeah. Is that fine? So this should be surprising because rho 1 and rho 2, the two myosin species, are not interacting with each other. Right? They were two myosin species. There is no interaction term between them, but they're interacting with each other via the elastic meshwork. And simply their local application of forces and unbinding mediated through this elastic meshwork drives this instability. So in fact, to linear order, one can actually show that the elastic power defined by that energy density is negative, meaning that the time derivative of this elastic energy is less than 0, meaning that at least to linear order, W is a Lyapunov function, and it drives the instability. There's some peculiar things that happen. For instance, that if in this unstable, when you get patches of high, let's say, where there's A-rich myosin and B-rich myosin there, in these patches where there is A-rich or rich in species 1, you see that there's a local increase in density as you expect. But there's also a smaller maximum of the other species. Okay, so which means that these segregated patches has a rho 1 which is colored in orange, but they're little tiny islands of rho 2. This sort of harks back to what Laetitia talked about in her lectures, okay? About coexistence of these. She had these gas bubbles in the MIPS condensed phases, right? Okay. So now in traditional, so we just done the linear analysis. We can ask now what is the contribution coming from nonlinear terms. And in traditional phase separation, what you would expect is that you start off with the homogeneous phase. Linear stability will drive it into this kind of bicontinuous phases if you have equal concentrations of rho 1 and rho 2 with a dispersion curve which looks like so. Okay. Eventually, as time progresses, nonlinearities kick in, and they'll drive it to a slower domain growth with the standard coarsening dynamics of T to the 1/3, and giving rise to large-scale domains such as these. This is what you would expect in ordinary segregation. Here, however, because these segregated patches are associated with contractile stresses, is applying contractile stresses, the region here is contracting more than the region there, because this is the higher contractility species and this is the lower contractility species. So indeed, if I create start off with a domain wall like so, okay, then domain wall given by phi, yeah, then what happens is that because of the enhanced contractile stress here and the back pressure coming from the outside, this peak here will rise and the width will shrink. Yeah, so rather than it expanding, the domain expanding forever, the domain grows for a while and then shrinks. Yeah, because this not only is segregating, but is also applying stresses in the medium. Is that clear? Yeah, yeah. Okay. So these domains, in fact, shrink after because of the existence of non-linearities, and this is what gives rise to finite time singularities. So one can actually calculate the way the shrinkage happens. As the shrinking happens, the peak of the phi increases to infinity and the width decreases to 0. Okay, and the way it increases, the phi increases to infinity, and concomitantly how the strain increases to infinity is of this form, and this can be obtained by doing a standard mass asymptotic calculation and the finite time singularity calculation to get this. And this is numerical. This is numerics which shows that if you plot it in this form, this shows a collapse. Okay, this is in phi, and this is in the strain actually. Yeah. So this is the finite time singularity that I was talking about. That segregated domains, because of this, so a linear description will tell you that there's a segregation instability. It will then grow to this bicontinuous like structures. But now nonlinearities kick in, and because of this nonlinearities, there's a compression, along the width of the strip. Okay, and as I think because it's a conserved order parameter, the height of the phi will increase. So it compresses and the local density gets enhanced. Okay, and one can analyze the shrinkage and the divergence of the amplitude of phi by this matched asymptotics and finite time singularity calculation it's using a dominant balance calculation to get this form. Yeah. So I don't know how to do that. I'm sure one can do it, and this would be an interesting thing to get. Yeah, I don't. What we can get is from a very simple dominant balance, one can only get these exponents and the scaling variable. If you have some ideas, this would be an interesting thing.
But Madan, this scaling will be valid in which regime? When x is large and t naught minus t both large? And
when x is going closer to x0,
yes,
and t is going closer to t.
Smaller.
Yeah.
Then t to the power 1/3 is usually you will get at late time regime, right? So
right, but that is for the growth.
yes,
right? This is the shrinking. So it's a different physics. This is coming from the dominant balance of the time derivative or the speed at which this thing and this nonlinear this B2 this B2 contribution whose origin is there.
it's not the usual.
It's not the usual. It's not the usual t to the 1/3. Yeah. Okay. So the analysis that we did here and the analysis that we did here and the numerics that we have here are done in one dimension. In 2 dimensions, one needs to be slightly more sophisticated. So this is and introduce anisotropies in the stress tensor. Okay, and only when you introduce anisotropies in the stress tensor do you get the same physics that I talked about. Okay. Yeah. Numerically solving the PDs. Yeah. By spectral methods. Yeah. Okay. We are having a lot of difficulty trying to do a 2-dimensional vector method. So again, any help would be much appreciated. I'll just show you this. So basically in 1D, you get the emergence of these of singular points. In 2 dimensions, these points become lines. And in 3 dimensions, they could be surfaces. Okay. For instance, this is a nice picture, an agent-based simulation by Francois Nedelik. Again, starting with the same, kind of initial conditions, you get the. Do you see the singular lines? Yeah. The singularities, of course, is suppressed by the steric interaction and so on. So it's not a real physical singularity, because the molecules have a finite weight. This is agent-based simulation. Yeah, Brownian dynamics. Yeah. Langevin dynamics. Sorry. Yeah. Okay, so I'll skip. Okay. Now, so what happens in maybe? Oh, how much time do I have? 5 minutes. Okay. Yeah. Okay. So now one can ask whether are there experiments to compare things with? There's recently a nice set of experiments by this group, which actually shows that if you trap these cells in sort of on a substrate with a patterned substrate, like this cylindrical patterned substrate, and then observe actomyosin, you see that there is dynamics of these lines. You see. Again, a lot of this needs to be analyzed. I mean, this is just someone else has done this experiment I looked at. But these are the kinds of experiments that one needs to compare this kind of theory with. Yeah. Now all the calculations that we did was on unbounded regime. Eventually, let's say if you do it in 2 dimensions, eventually, you'll get this singular structures only if sigma and n being the normal to this patch. Yeah. Yeah. That's a good thing.
It looks like there aren't too many more than 3 coordinated things there. Is that? Or am I just mistaken?
I think that was
just
the
picture.
Yeah, that was just a
okay.
Yeah. Oh, in the simulation. Yeah. That had only right. But in images, you find more things in real images. So yeah. So what I wanted to say here was that this singular lines only exist in the regime where the normal component of the stress tensor is larger than the tangential component. But eventually, so it'll form the structure, shrink to a line, but eventually contractile stresses from here will destabilize this line. Okay, and it'll make it small blobs. Now, in order to stabilize lines, you need to have the surface anchoring. Remember, that's what I told in the first slide, that these stress fibers are only stable when cells are adhered on a substrate where there is surface anchoring. And indeed, when you put surface anchoring, we have done this work. When you put surface anchoring, you get these web of lines which can be stabilized. Okay, in a real cell, these surface anchoring is provided by these molecules, focal adhesions and adherence junctions. The moment you get rid of these molecules by detaching the cell from a substrate, you lose these pattern of lines. Okay? Yeah. So, and this is an interesting. So one can write mechanical, equations for mechanical stability of these lines at junctions. There are these Young-Dupré equations, etc., etc. You can derive those equations from the kind of analysis that I talked about. Now, what's interesting here is that this kind of schematic is true for this particular realization of these anchoring. The moment I move or change this anchoring to slightly different positions, the whole web will change. Okay, so this is a fragile system, very similar to what you see in granular media. But here, unlike in granular media, where the fragility is held by compression, here it is held by tension. Okay. Yeah. So this indeed is is an example of an active fragile material of force chains, but the force chains are held by tension and not by compression. Okay, so I think I'll end there. And there's a lot more work that we need to. Oh, I haven't told you about excitability because I don't have the time. But I'll just say that there are excitable waves here. There is a regime in parameter space where you get traveling waves, and this can be studied analytically. And something called swap. So that's a traveling wave solution. And this is a swap phase. You can see it's like a standing wave. Okay. And in other regimes of parameter space, you get this funny coexistence, which we haven't understood. A coexistence between traveling waves and swap. Okay. So you can see that it's swapping, and after some time it will travel. And the swapping will cross over to a travel, and so on. And then it often comes back, and so on. So there seems to be a temporal chaotic transition between swap phase and traveling phase and back. So something that we haven't understood. But, this is all in 1D. Yeah. And though the linear stability and this linear stability, this is an analytical calculation. So this works in any dimension. Okay, our next goal, future goal in some time is, remember, the cytoskeleton has two kinds of structures. One is called actin, which is what I described earlier, and the other is called microtubules. The difference between these two, apart from their appearance, is that these actin holds tension and microtubules hold compression. Okay, so now you have a combination of compression and tension. And this potentially gives rise to a phenomena called tensegrity. And I don't have the time here to do this, but these are amazing material structures, first proposed by Buckminster Fuller, of having these kind of structures, rods segregated from these rubber bands, and this structure is stabilized because of tension in these rubber bands. The moment you cut these rubber bands, the whole thing collapses. Yeah? And there has been a proposal by Ingber that the mechanical properties of the cell should be described by such tensegrity kind of models. And what we would like to hope to do is to provide a dynamical theory for tensegrity. So that's the goal. Okay, so I'll end to say that I discussed this non-equilibrium force patterning in the cytoskeleton, mainly from the actin cytoskeleton. Gives the fact that they were force generators and force sensors, both force generators and force sensors, was crucial in describing the dynamics towards these singular structures. So what we found is that there's an emergence of stress fibers or singular lines, which are stabilized by surface anchoring, which coexists with excitability. And what we are hoping to do in the future is to come with a dynamical theory for tensegrity. And these are the people who have done the work. Ayan is a very extremely talented postdoc working in our group, and Saptaresh is a PhD student at the Simon Center. Okay. And this is earlier collaborations on similar ideas of cells embedded in tissues with Thomas Lequier's group in Marseille. Okay. Thank you.
Questions?
So I have a question about the two instabilities, this segregation and the contractile. So does the contractile instability already exist within the non-segregated phase? Like if you compress it a bit or some fluctuation happens, then lines will form, or?
No, so the contractile instability is a different phase. It happens at a different value of the effective bulk modulus.
So you can have segregation without contract.
Yeah. But it's interesting, you know, because as pointed out by Kabir, that the relationship between the renormalized elastic modulus and phi is a local quantity. So, you know, you could have in one region something that is approaching right? Approaching a contractile instability. So within the segregation region, what happens within the patches approaches a contractile instability without ever getting into it. So, okay, so from the theory, let's say I do linear stability analysis. Then the interesting phases do occur in a large range of parameter space. Comparison with experiments. So some of these phases, for instance, traveling wave phase, etc., happens in a very narrow region. It happens typically as a boundary between the oscillatory phase and the contractile instability. So that's a limited. About extracting these parameters from experiments it depends on what kind of experiments. The best way to make such a comparison would be on in vitro systems. So remove things outside the cell. Those experiments haven't been done as yet.
Okay, so maybe since we're over time, we can continue the discussions over coffee. So let's thank both Rahul and Madan.